Assalamualaikum and a very good day. My name is Nur Shasha Izzati binti Nasaruddin, and today I would like to present my case study entitled Screening for SARS-CoV-2 spike gene in the genome of potential mammalian hosts. Firstly, for background of study, SARS-CoV-2 or SARS-CoV-2 is a novel coronavirus which causes severe acute respiratory syndrome. It is responsible for the spreading of coronavirus disease 2019 or well known as COVID-19 all around the world. While for spike gene, it is located at the surface of the virus and allow binding to the host cell receptor. It promotes the entry of the virus into the cell, thus infecting the possible host cell which is from the mammalian host. As you can see on the figure, this is the structure of SARS-CoV-2 and we can clearly see there are spike gene all around it. Next is mammalian host. It has the ability to engage with the spike gene by the binding of spike gene into their ACE2. Example of hosts that can potentially recognize spike gene are Indonesian tapi, which is on the left figure, and Malayan pangolins, which is on the right figure. As for the bioinformatics, it is a technology that coping with infections by investigating genetic sequencing and seeking structural models of potential molecular targets present in SARS-CoV-2. There are three problem statements for this case study. Firstly, the interaction between spike gene of SARS-CoV-2 with mammalian hosts was less studied by most researchers, although spike gene is the main role in the infecting process of COVID-19. Secondly, the infection of SARS-CoV-2 from camel and other mammalian hosts, which can possibly assume that mammals did share the same spike gene. And thirdly, any similar spike gene that exists interact with other mammalian hosts needs to be studied in order to achieve better understanding regarding interaction of SARS-CoV-2 spike gene with mammalian hosts. There are few significant of this study. Firstly, it will be beneficial to public and will contribute to the field of research and science in the origin of COVID-19. Secondly, if the similar spike gene that exists in another mammalian host is successfully obtained, this mammalian host can be further used in the clinical field. For example, it can facilitate any further preclinical analysis of vaccines and therapeutic agents by testing therapeutic agents and vaccines towards these mammalian hosts. Therefore, the pandemic of COVID-19 can also be handled efficiently and effectively when effective vaccine and therapeutic agents has been successfully tested. There are a total of three objectives of this study. Firstly, to retrieve the homologous SARS-CoV-2 spike gene in the genome of mammalian host database using BLAST program. Secondly, to align SARS-CoV-2 spike gene retrieved from mammalian host database using sequence alignment tool. And thirdly, to investigate the evolutionary relationship of spike gene from mammalian host database using phylogenetic tree software. As for the literature review, the role of spike gene in COVID-19 infection is they intervene the virus engagement with the host cell surface receptors and their subsequent fusion to infect the host. Interaction between the spike gene and its receptor will begin the replication of coronavirus. And it also forms homotrimers that will bind to the viral surface. As you can see in the figure, this is the conserved motif of S2 subunit of spike gene. For example, STSQ, ICGD, and PTNF. As for the relationship of SARS-CoV-2 with mammals, SARS-CoV-2 falls into the subfamily of coronavirus which is beta coronavirus. Therefore, it conserves an ability to cause infection towards a wide range of mammals. Some mammals that potentially recognize spike gene are dog, cat, and golden hamsters. This finding indicates that these mammals have the possibility of getting infected by SARS-CoV-2, although the infection disease might not be as severe as infection in humans. Next is phylogenetic analysis of SARS-CoV-2. SARS-CoV-2 shares 89% of its nucleotide identity with bad SARS-related CoV, and it shares 82% of its nucleotide identity with two types of human SARS-CoV. The 2019 nCoV was associated with lineage beta-coronaviruses in phylogenetic tree that was built by using the amino acid sequence of the four structure gene, which was spike, envelope, transmembrane, and nucleocapsid. There are four research methodologies for this study. Firstly, primer selection. And for primer selection, conserved region of SARS-CoV-2 spike gene from human was chosen from genels, and the chosen conserved region was S2 subunit of spike gene. And during the process, after the nucleotide sequence was obtained on the respective location, the sequence was pasted on the ORF Finder website to translate the sequence, thus obtaining the amino acid sequence of the S2 gene. Secondly is database searches. The genomic database of COVID-19 spike gene from mammal was obtained by using the GenBank website. Thirdly is sequence alignment that was being conducted by using Cluster Omega program, and this program aid in aligning multiple nucleotide or protein sequence efficiently, and the format that has been used for amino acid sequence was in FASTA format. Fourthly, phylogenetic analysis was being conducted by using MegaEd software. Phylogenetic tree of the small mammal genome sequences with the primer sequence was created, and the relationship of spike gene from human to other mammalian hosts can be identified. Next, I will talk about my results and discussion. Firstly, primer selection for SARS-CoV-2 spike gene. There are a total of 7 primers that were selected from Southeast Asian patients, which is 1 from Wuhan, 2 from Malaysia, and 2 from Indonesia, and another 2 from Singapore. Secondly, database searches for SARS-CoV-2 spike gene. This was conducted by using BLAST program from the gene bank, and the focus for this study is on mammals in view of the fact that SARS-CoV-2 mainly infects mammals, and not only that, it also has the ability to infect a wide range of mammals, which includes humans and animals. There are a total of 18 mammal species that has been infected with coronavirus, and its genomic database was obtained. In the table below, I stated the species and the genomic database, and it also continue on the next slide. For the next discussion and results, as you can see on the slide, this is the sequence alignment of SARS-CoV-2 spike gene from mammalian hosts and primers from human. And as you can see in the figure, the symbol of star indicates that the amino acid has fully conserved, while the colon symbol indicates that there are conservation between groups of amino acids with strongly similar properties. However, the full stop symbol indicates that there are conservation of amino acids but with weakly similar properties. Therefore, from this sequence alignment, it can be observed that there are similarity from 4 amino acid sequences such as QIPF and VINQ. And it can also be proven that S2 subunit of spike gene is highly conserved. The next result is regarding phylogenetic tree. As you can see on the slide, this is the phylogenetic tree of SARS-CoV-2 spike gene from mammalian hosts and primers from human, which is a total of 18 spike gene from mammalian hosts and 7 primers from human. Therefore, there are a total of 25 species in this phylogenetic tree. From the phylogenetic tree that I've shown just now, phylogenetic analysis that can be made are all of the species from this study came from the same ancestor, but level of relatedness is different from one another. Firstly, spike gene from bat species has 100 bootstrap count with human SARS-CoV-2 isolated from Singapore, which has been proven by researchers that stated the human SARS-CoV-2 showed around 93% sequence identity with bat SL-CoV ZC45 and bat SL-CoV ZX C21. Secondly, S2 of SARS-CoV-2 from pangolin species has 87 bootstrap support with human SARS-CoV-2 isolated from Indonesia, which has been proven in journal that stated spike gene of pangolin CoV 2020 and human SARS-CoV-2 have 89.9% to 92.4% sequence similarity. Therefore, it can be observed that the most ancient coronavirus are bat coronavirus HKU41 and bat coronavirus HKU51. As referred to Chan, these both coronavirus are located at the lowest position of phylogenetic tree, which is the same with the one that I got from my phylogenetic tree. In conclusion, there is an evolutionary relationship of spike gene between mammalian and human hosts. However, the exact origin of SARS-CoV-2 spike gene remain unknown because not enough information of spike gene in their mammalian hosts. Therefore, my recommendation is further investigation needs to be conducted in order to obtain more information of spike gene in their mammalian hosts in order to identify the origin of SARS-CoV-2. For example, more animal sampling can be done to determine the natural reservoir of SARS-CoV-2. Before I end my presentation, this is the video I found on YouTube that explains what is spike gene of SARS-CoV-2.
SARS-CoV-2 virus has a couple of proteins on the surface that we can utilize to develop serology assays. One of those proteins is the spike protein. The spike protein consists of 2 subunits, an S1 subunit and an S2 subunit. On the S1, there is the receptor binding domain, which is responsible for the ability of the virus to bind to the human cell, infecting that cell. Antibodies against the receptor binding domain have been demonstrated to be neutralizing to the virus. In other words, disarming the virus's ability to bind and then further invade that cell.
Lastly, these are the references that I used to complete my final year project, which is a case study. Therefore, I end my presentation. Thank you.